Now we are going to take the final step, which involves the regeneration of RUBP. As you know, RUBP was the compound which underwent carboxylation and it got converted to 3-phosphoglyceric acid. Now we are going to see that RUBP which was utilized and it got converted to 2 molecules of the phosphoglyceraldehyde, it needs to be regenerated so that the more carbon dioxide that is coming, it must combine with something which is regenerated. Otherwise, the RUBP would be used up and the plant will have to provide it. So, for providing it, the regeneration is necessary, and we are going to see how we are going to get back that ribulose 1,5-bis phosphate that came in the beginning and got carboxylized. You know that fructose 6-phosphate was being formed when there was glycolytic reversal, and along with that we had that DHAP that was also being formed. We have the reaction fructose 6-phosphate get combines with 3-PGAL. This is glyceraldehyde 3-phosphate. As I told you in the beginning, it could be written as 3-PGAL. This fructose 6-phosphate, it combines with glyceraldehyde 3-phosphate and it leads to the formation of 2 compounds, erythrose 4-phosphate and xylulose 5-phosphate. These are the compounds that would be formed in the presence of enzyme transketolase. Now, this is little bit confusing. You would think that where from these compounds have come. I would suggest you just to keep it little bit simple, you just memorize the names instead of getting into the details of the chemistry of these compounds. These 2 compounds, this is a 6-carbon compound, this is a 3-carbon compound. Both of them are forming the compounds erythrose 4-phosphate and xylulose 5-phosphate. Both of these come together and they form these 2 compounds. Now, out of these, the erythrose 4-phosphate, this particular compound, it comes and combines with isomerase of this 3-PGL, that is 3-DHAP. Firstly, fructose 6-phosphate would combine with the glyceraldehyde that was formed out of 3-PGA. Secondly, the same isomer which was formed out of this would combine with the outcome, that is erythrose 4-phosphate. In return of this reaction, we get pseudoheptulose 1,7-diphosphate, which has 2 phosphates over it. We have a compound known as pseudoheptulose. Okay, it would be a 7-carbon compound. Now, next what we have is pseudoheptulose combines with water in the presence of phosphatase and gets rid of one of the phosphate which was present at 1 and 7 position. It combines with water, there is phosphate removal and we have that phosphoric acid. This is formed pseudoheptulose 7-phosphate. Please remember the reactions, memorize them, don't get into details. Just keep in mind that fructose 6-phosphate that was to form the glucose, it comes and combines with glyceraldehyde 3-phosphate. The outcome of this reaction combines with another isomer of this glyceraldehyde 3-phosphate, that is DHAP. The outcome of this particular reaction combines with water and forms pseudoheptulose 7-phosphate. Now, this pseudoheptulose 7-phosphate combines again with this 3-PGAL and forms xylulose 5-phosphate and ribose 5-phosphate. You have 10 carbon compounds and both of them they form 5 carbon compounds. Now, this xylulose 5-phosphate would get converted to ribulose 5-phosphate. Xylulose would isomerize in the presence of epimerase to ribulose 5-phosphate. This ribulose 5-phosphate would combine with 1 ATP, and as we wanted 2 phosphate groups in the ribulose, this ribulose would get converted to ribose in the presence of isomeric reaction, which I have skipped over here. Just remember, this ribulose 5-phosphate would get converted to ribose 5-phosphate, which would be an isomeric reaction. Now, this ribose 5-phosphate would combine with ATP to form ribulose 1,5-bis phosphate in the presence of enzyme phosphoribulose. Do keep in mind that I have skipped one point over here where ribulose would get converted to ribose, and this is an isomeric reaction. Now, it is quite confusing. We just started with 2 PGA compounds. Now, we are getting PGAL, DHAP, PGAL again and again, and where from they are coming? For that understanding, this representation, that is simple chemical reaction representation, would not be enough. For that you need to see the graphical representation, where we get an idea that all the biochemical processes, they are not taking place in vacuum. All the things are going hand in hand. If at one side you are getting PGA that is being generated out of one reaction, that reaction would not be in vacuum. That means it begins at one stage and it ends at one stage. What could be beginning of one reaction could be end of the other reaction. Now this is a confusing statement. For understanding this particular reaction in a better way, we need to see how Kelvin cycle takes place. And when I say Kelvin cycle, then that means we are going to see how the cyclic process is taking place. So for that you need to see the next lesson.